So to focus on the M phase, mitosis, which is the division of the nucleus. In mitosis, daughter cells have the same number of chromosomes as the starting cell. The daughter cells are your ending cells. So you have a parent cell and then daughter cells, and they have the same number of chromosomes as the starting cell. And there are 4 phases: prophase, metaphase, anaphase, and telophase. I know it as PMAT. And where this actually takes place in your body is everywhere except your reproductive organs in order to produce sperm and egg. Your reproductive organs do go through mitosis to create more reproductive organs, but not to produce your gametes or your sperm and your egg. So prophase, we're going to go through each phase now, and this is where the whirlwind of fun starts. You have your prophase, where your DNA turns into chromosomes, which, if you remember, chromosomes are coiled up DNA. So the DNA that's duplicated due to the S phase start to coil up to become a chromosome. And the reason for this is because the nucleolus and nuclear membrane start to break down. So you want your DNA coiled as much as possible to protect it while your nuclear membrane breaks down. And this is where chromosomes can be seen by the light microscope. So if you look at the picture on the top, you'll see that ball, and those are your chromosomes. And your centrosomes appear. In animals, they're known as centrioles, and go to opposite poles. And this is the centrosomes and centrioles produce your fibers. And there's spindle fibers, which there are 2 different types of spindle fibers: kinetochore fibers and polar fibers. Kinetochore fibers are the ones that are going to actually move your chromosomes, so those fibers go and attach to your centromeres, and your polar fibers attach the centrosomes to each other to maintain that shape. So these fibers are really important, and especially the kinetochore fibers. If you think kinetochore, kinetic energy, movement, those are the ones that are going to move your chromosomes. So the next phase, your kinetochore fibers, because those are the ones attached to the chromosomes, move your chromosomes to the center of the cell. Not always in a perfect line, but pretty good and pretty easy to see. And I always think meta-middle, and it looks like a basketball. That's what I always think. Now the next phase, if you remember PMAT, is anaphase. Anaphase, the chromatids are actually separated at the centromeres and move towards the opposite poles. So those kinetochore fibers are pulling, basically play a tug of war with your chromosome and pull them apart and separate them. This is why it's so important that those DNA strands that were replicated had stayed together, because if they were moved to different sides at random, one cell might end up with DNA for 2 of the same strands of DNA, and then the other cell wouldn't have the DNA for that. So if it had a certain gene on it, like arm, having 2 arms, you wouldn't have 2 arms, and that's where problems could occur. Problems can still occur because if the chromatids don't separate, one cell gets 2 of the same chromatid and the other does not get any of it, which is a problem. But the fact that they're attached decreases this problem, because if they're just at random every single time, they will randomly separate and you don't know what you end up with in each cell. So that is anaphase, and you'll see the picture above. You can see the 2 lines because they're being pulled to the opposite poles. And then telophase is where the chromosomes start to uncoil and go to the chromatin, or uncoiled state. So they're starting to get harder to see. The spindle fibers start to break apart, and the nuclear envelope starts to form, and the nucleolus starts to form. And you'll see there's also a start of a pinching on the side, and that is our next phase. So that's the end of mitosis, which is nuclear division. The next phase, cytokinesis, is where you break apart your cytoplasm or separate your cytoplasm. So in animals, because we have the cell membrane that is easy to move, it pinches inward between the 2 poles and creates a cleavage furrow. So that is a picture of cytokinesis in animals. And then in plants, because they have that cell wall, which is rigid, they actually have those vesicles that fuse and form a cell plate. So it's basically creating a wall in between 2 classrooms, and that makes 1 classroom 2 classrooms. Makes 1 cell, 2 cells. So that is how you end up with 2 cells. So if you look at these stages, you can guess what stage each is at. And if you guessed those phases, you got them correct. Good job. Now if you look at this is less specific, but you can figure out how many different phases are there, and they're actually all there. If you got them, good job, because you're going to be doing a lab where you have to figure out what phase various things are in. So if you can tell, the metaphase that is being pointed to, as you see, it's not a perfect line, but it's actually pretty good for a metaphase. So the results of mitosis, if you follow that all the way through, and you've made it, the life of a cell that has divided, you end up with 2 identical cells, void of any problems that occurred, which are known as daughter cells, and they have the same number of chromosomes as they started with. So this is similar to cloning. Actually, this is cloning. You're cloning your cells. Now, that turns into a whole another issue of whether we should clone people and all of that, because stem cells are for cloning. If you know what a stem cell is, it's a cell that has no specialization and it can become anything. So if you think of the possibilities of if we can control mitosis and figure it out, well, if we have a cell that can become anything, then we can take that cell, make it go through mitosis, and then we would end up with, and then force it to become whatever we want it. And you get a heart, and you'd get a kidney, and their possibilities are endless. It causes slight problems ethically and all of that, but those are thought processes and how mitosis actually can kind of create something. So how this actually is controlled in your body, it is controlled by hormones, which are known as cyclins. Cyclins control the cycle. You have 2 types, the G1 and the mitotic cyclin. G1 is found in the G1 phase, mitotic cyclin is found during mitosis. And they basically make sure the cell only divides when necessary and does not get stuck in a stage. Because if it gets stuck in a stage, then that cell is pointless at that moment. And if it's dividing too fast, you could end up with a tumor. And that's actually what cancer is, or any tumor is cells not noticing when they should or should not divide, because generally it's how much pressure is around them. If there's tons of cells around them, they don't need to divide, but if cells are dying off around them, they're like, okay, we need to divide, start dividing. But a tumor is when the cell does not know how to stop dividing and just rapidly divides. So that causes problems. There are checkpoints to make sure that there are no problems. Obviously does not always work, and if there are problems, your cell cycle goes into arrest. It's like it goes to prison and can't pass go, can't collect $200, goes into jail, and actually creates inhibitors to stop the cyclin system. So your cell can actually control this, but again, there are problems that can arise, and that's generally due to mutations in your DNA. And that's because this is all controlled by your DNA. But there are points that the cell can notice if something's wrong. If it goes into arrest, it can stop the cell cycle, and it also often just kills those cells off and then creates more. So it's not that big a deal. But if your cell is not checking itself properly or not paying attention to those cyclins properly, then that could arise in a problem. So how this knowledge actually can help is knowing the speed helps diagnose cancer and other genetic disorders, and can help research and find cures for genetic disorders. Now, because we know more about chromosomes and cell division, we can look at what are known as karyotypes to also determine genetic disorders. Karyotypes are pictures of your chromosomes. Now, you're supposed to have 46 chromosomes, 23 pairs, and those pairs are your homologous chromosomes. So a normal human karyotype, you end up with your 23 pairs. Your last pair are your sex chromosomes. So on the left, you've got a male, XY, and on the right, you've got a female XX, and the way they pair them up is by size and shape of your chromosome. So the left, obviously, they've made it easier for you because they're color-coded, but on the right is actually what they look like. So they take a picture of your chromosomes in prophase and then pair them up and figure out what goes where. And then they can determine what's wrong. If there's something wrong, they can do this very, very early in a pregnancy to determine if there will be a chromosome just disorder. And the first one, you'll notice that there is a third 21st, which is known as trisomy 21, which is Down syndrome. Then the next one on the top, on the right, you'll notice that there's only one X chromosome, no Y, and no second X. And then the bottom left, you'll notice that you have two X's and a Y. And then on the bottom right, you've got the three X's, triple X, so super female. So you'll notice that those are all chromosome disorders or DNA genetic disorders, which usually resulted from chromatids or chromosomes not separating properly. And more likely it's homologous chromosomes not separating properly during meiosis, because these usually occur during pregnancy, or during fertilization actually, and then discovered during pregnancy. So these things can actually occur. So those are your answers to your abnormal karyotypes. And something that you probably have heard of is Down syndrome or trisomy 21, because you have an extra chromosome. So extra chromosomes actually cause problems. And when we talk about meiosis, which is our next fun adventure into the life cycle of a cell, and a little story of how cells live, then you will find out how that more specifically can occur. But for now, just realize those are pictures of chromosomes, and now that you know that they're available in prophase to be seen by a light microscope, you can easily diagnose problems. And now that we know how many we're supposed to have, all of that, we can diagnose problems a lot easier, which is a good thing. So hopefully you survived the whole story of a life cycle of a cell, and when we go into meiosis, you'll see a lot of similarities, and you'll now know how you went from one cell to all of your different cells in your body.